All right, so this is a video about the endomembrane system. And the endomembrane system, I just want to talk about what it is because the first thing is I can define it. So let's look at what the words mean. So endomembrane, endo means inside, and membrane just tells you it's made of membranes. So we're looking at something made of membranes that is inside something else. Well, since we're doing this in Unit 2, which is the cell unit, I bet we're talking about it's inside the cell. All right, so we're looking at things made of membranes inside the cell. All right, that's the first word. System. System is anything made of multiple parts that work together to accomplish a new task. So if you think about your body systems, like your respiratory system or your circulatory system, cardiovascular, whatever you want to call it, your heart can't do really a whole lot by itself, right? If you took a heart out of a person and put it on a table, it could pump and squirt blood everywhere, but it's not going to get blood to my toes. But when you put the heart with the arteries and the capillaries, and then the veins and the valves and all that stuff, then you've got multiple parts. They're all working together, and they are now accomplishing a task that each individual one could not accomplish. So while my heart can't pump blood all over my body without the veins and arteries and all that, when you add them all together, then it can accomplish that task, and it can get blood to my toes. And so that's what we're looking at here. We're looking at something, these pieces of the inside the cell made of membranes, that's your multiple parts, and these are going to work together to accomplish a task, and that task is going to be transporting lipids and proteins around the cell or even out of the cell. And so the next one says, I can tell which organelles are members of this. That is going to be your nucleus, because it's providing the DNA to make everything. That is also going to include your rough and smooth ER. Because they are producing the proteins and the lipids that are leaving. Also your Golgi, because it's packaging everything up and shipping it out through vesicles. So vesicles are a big part of it. Your cell membrane is a big part for anything that's leaving. And then lysosomes are actually a part of it as well, and we'll talk about that in just a second. So I can describe the movement of molecules through the endomembrane system. So over here, let's say that we have the rough ER or the smooth ER, okay, making some protein or some lipid. It's going to send it to this guy. Hopefully you recognize that. I know my art skills aren't great, but that one's not too bad. And that's the Golgi apparatus or Golgi body. And so what the Golgi is going to do is it's going to modify that protein or lipid. It's going to finalize it if it needs to have a hydroxyl group or a carboxyl group, or, those kinds of functional groups or something like that added to it. If it needs to be folded a certain way to make the structure right so the function will be right, then the Golgi is going to do all that. It's going to kind of finalize it. It's then going to package it up into a vesicle, and it's going to ship it wherever it needs to go. And so you see some vesicles budding off here. There's one that's getting started. There's one that's almost done. These guys have already popped off. They're moving towards the membrane. Here's one that's already fused with the membrane, and it's going to open and dump all of its stuff outside. And so that would be the movement of molecules. So from the rough and smooth ER, usually they do go through vesicles here to get to the Golgi. Then the Golgi does its boom, 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 and then it's going to put it back in another vesicle and ship it to the cell membrane. Or some of these vesicles may be going to other organelles. Some of these vesicles may be full of a protein that is a hydrolytic enzyme, and that is going to become a lysosome. So some of these vesicles can actually become something else, like a lysosome. Or it could come over here, maybe here's a lysosome. It could fuse with this lysosome and dump its contents, as they fuse and then open up. So it would kind of look like this, and it would dump those contents into that lysosome. So if the lysosome needs more enzymes, if it needs to be recharged, so to speak, it can do that. I do have this picture up from the PowerPoint because it's a much better picture than what I can draw. So you can see things coming from the rough ER to the Golgi, getting fixed or whatever, and then sent out the other side of the Golgi to the cell membrane to be released through exocytosis, which you now know what that is. All right, so I can describe the movement of molecules through that system. So we did that. And then the last one is I can explain why the location of structures in the ER, Golgi, or vesicle is the reverse of the location on the membrane. So let's kind of zoom in and let's show that. So let's say here's a piece of the Golgi, and let's say there's a receptor that I need to put on my cell membrane. Okay, so this is a whole nother cell. What that Golgi is going to do is it's going to package that receptor into a vesicle. Okay, zoom in a little bit so you can see my receptors. Not that I'm doing a very good job of drawing them. And then that's going to go all the way to the cell membrane, and I should have, let me use a little white out here, get rid of some of my cell membrane. I should have left a gap in my cell membrane, but, you know, not really, but because what's going to happen is my vesicle is going to come over here to my cell membrane, and this receptor that was on the inside, so imagine this right here is the inside of that vesicle. Okay, it's fused and it's opening up, and so then when my cell kind of straightens out, my receptor is now on the outside. Okay, by contrast, let's get rid of that. All right, let's move to this little blank space. Let's say that we had something that was on the outside of the Golgi. Okay, and so then when I make my vesicle, it's going to be on the outside of my vesicle. And then when my vesicle fuses with my cell membrane, this inside is this inside here, which now becomes the outside. So anything that's on the outside of my vesicle, this is the outside back here. So now that whatever, receptor, protein, whatever, is going to be facing inside the cell. And so basically wherever it is in the Golgi or the rough ER or the vesicle, if it's on the inside, when you fuse it with the cell, it's going to be on the outside. And if it's on the outside, when it fuses with the cell membrane, because the outside of the vesicle becomes this part of the membrane, then it's going to be facing inside the cell. So it is going to be in reverse. I hope that helps. If you have any other questions,